I should know. No? Okay. So let's go on. Yes, please break it. We don't need it. I always say that to you guys. Huh? But it's okay. You will see that in YouTube and you start laughing. Huh? Ah, you fell off. It's still recording. Let me turn it off. So, and then, okay. Now it's all crooked. Okay, we got it fixed a little bit. Okay. No? Still recording. Okay. Well. Okay, let's go. Proteins accounts for more than 50% of the mass of the most of the cells. Here, let me grab these cells. We need them. I need them. You need them. Let's break into it. So, what am I saying in that diagram? That this cell, about 50% of it is mass. It's weight. 50% of it is protein molecules. More than 50% is protein molecules. See this here. This cell, this is a plant cell, about 50% or so of the weight of this cell is protein molecules. So they make a big bulk of a cell. Some proteins speed up chemical reactions. Other proteins function includes defense, storage, transport, cellular communication, movement of structural supports, and you will see some of those. Enzyme activity. Enzymes are made up of protein molecules, and we will not talk about them now, but next material, next exam, you will see them. We will talk about them. Defense, they make antibodies. If you, God forbid, you come down with a cold or influenza, that's what your body will make a Y-shaped antibodies and then fight the virus, what happened. Storage, eggs, okay, so storage of nutrient and in there. Transport proteins in the cell membrane, which we haven't studied it yet, but we will. It allows other molecules to go across. So this is a protein molecule within the cell membrane. What else? Hormonal proteins. Hormones. Insulin is a protein molecule. Growth hormone. You've heard about it. At night when you're sleeping, you release a lot of it. Growth hormone. So it's protein molecule. Receptor proteins on the surface of the cells. We will talk about this later on, next chapter material, that they have. The cells have receptors, and then when neurotransmitters is released, then the receptors react. Contractile and motor proteins, your muscle contractions. When you are allowed to do this, Sean, is that right? When you're allowed to do this, you are using your proteins in your body. Structural proteins, your cartilage, your nose, your ear, your joints, they're all protein. Okay, that's what you're talking about, structural protein. Okay, they have a few animations. I hope it will not take long. Let's quickly look at it. I think there are 4 or 5
proteins, like the motor protein shown here, can move parts of a cell. Other contractile proteins, working together in muscle cells, can move a whole animal. The immune system makes defensive proteins called antibodies that bind to invaders, such as the virus shown here, and mark the foreign objects for destruction. An enzyme is a protein that changes the rate of a chemical reaction without itself being changed into a different molecule in the process.
Sorry. Anyhow. Let's go.
In cells.
Yeah, well, we'll talk about enzymes a little bit.
Gene regulatory proteins bind to DNA in particular locations and control whether or not certain genes will be read. This allows cells to become specialized for different functions and respond to changes in their surroundings. Signal proteins include hormonal proteins that help coordinate an organism's activities by acting as signals between cells. For example, insulin, a hormonal protein secreted by the pancreas, signals in animal cells to take in and use sugar. The hormone receptor is also a protein. Receptor molecules bind to signal molecules and can then emit second messengers which trigger changes inside a cell. Receptors are thus important links in the system of communication among cells. Some signal molecules, such as hormones, are also proteins. Sensory proteins detect environmental changes such as light and respond by emitting or producing signals that call for a response. Storage proteins stockpile building components that cells can use to make other proteins. Storage proteins in seeds provide raw materials used by the developing plant, unless an animal eats them first. Ovalbumin, the main substance in egg white, serves as a storage protein for developing chick embryos. Structural proteins have many functions. Like tent poles and ropes, they shape cells and anchor cell parts. They may serve as tracks along which cell parts can move. They bind cells together, making organized units such as muscles, ligaments, and the tendons that bind muscles to bones. The silk of spiders and the hair of mammals are also structural proteins. Transport proteins carry molecules from place to place. The example shown here allows certain solute molecules to enter the cell. Transport proteins carry molecules from place to place. The example shown here allows certain solute molecules to enter the cell.
Okay, so that's it. Most of these guys, most of what you saw in these little clips, we will talk about it for the rest of semester. Okay, but this first exam, just get familiar with what proteins are. Proteins are, this is a monomer. That monomer is called amino acids. And we talked about it last time. Quickly, I'm going to go over it so you know everything about it. And amino acid has a central carbon. He's calling it alpha carbon. During the exam, I called it central carbon. I didn't go by alpha carbon. Okay, so central carbon, attached to the central carbon is an acid group, which we call carboxyl, same thing, carboxyl group. And then you have amino group, okay? You have a hydrogen, and then you have the R group. The R group is the radical. R stands for radical group. It means they are all different, all 20 amino acids. You and I are made up of 20. In the nature, there is about 120 different amino acids, but we human are made up of 20 different amino acids, which are listed right here in your textbook. Quiz question today I asked, which one of the following is amino acid or which one of the following is not amino acids? So glycine is one. Please learn how to pronounce them. You need them for the rest of your life. Glycine, alanine, valine, leucine, isoleucine. So these two are very much close to each other. Methionine. Methionine is one of those amino acids. There are two amino acids that have sulfur. Methionine is one of them. Okay, methionine is one of them. Phenylalanine, tryptophan, proline, serine, threonine, cysteine, right here. You see that one? You will see this for the rest of semester a lot. Cysteine. As I said, if your hair is curly, because you have a lot of cysteine. If your hair is not curly, you don't have much. You want to make your hair curly, they go to the beauty shop and they add a gel or whatever that has cysteine, that has sulfur. Okay, not cysteine, that has sulfur, and they iron it, then your hair becomes curly. When you have a curly hair, you want to make it straight, use iron to break these disulfide bond, which I will talk about it. Okay. Then tyrosine, you'll see this again in the rest of semester. Asparagine, glutamine, and then you have these acidic. There are two amino acids that are acidic: aspartic acid and glutamic acid, and three amino acids that are basic: lysine, arginine, and histidine. Yep, that's histidine. Okay, so these are the 20 amino acids. I'm hoping you would know the 3-letter abbreviation, 1-letter abbreviation, and so on and so forth. For rest of semester, we do need them. You do not need to know the structures. I don't expect you to know the structures. You're not taking a biochemistry course. Whenever you take biochemistry later on in your life, there you do need it for the rest of the semester if you take a biochemistry course. But you don't need it here now. We just need the names, that you be familiar with the names and the 3-letter abbreviations. Brian, listen, okay. Non-polar side chains are hydrophobic side chains. These are, do not, again, I don't expect you to know the non-polar and polar amino acids. Your textbook emphasizes on it. It gives you examples and so on and so forth. Again, I'm not too worried about it. And here are the polar side chains, hydrophilic, the one that they love water, so they can interact. What hydrophilic means, it means they can interact with water. Okay, and hydrophobic ones, electrically charged side chains, and hydrophilic again, these are the amino acid they like to act water. This is the nomenclature of protein molecules, amino acids. You all know what I mean when I say nomenclature, you all know what I mean. Okay, the naming system of it. I gave it to you for carbohydrates, and here they are for polypeptides, amino acids. So peptide bond, and I will talk about that here in a minute. Hang on. What is a peptide bond? When you attach 2 amino acids together, the bond between the 2 amino acids is called a peptide bond. Right here. This is called a peptide bond. Okay, I will talk about that. A polypeptide is a polymer of amino acid. A dipeptide, when 2 amino acids attach to each other, is called dipeptide. When 3 amino acids attach to each other, is called tripeptide. So the term tripeptide exists. Does the term tri-glucose or trisaccharide exist? Anybody? No, I never heard of it. In my life, I never heard of trisaccharide. You've heard of disaccharide, right? Huh? You've heard of disaccharide, but I never heard of trisaccharide. But we do have tripeptide. It means 3 amino acids attached to each other. After that, anywhere between 4 to 10 is called oligopeptide. And between 11 to 50 is called polypeptide. Between 50 to infinity, then it's called protein molecule. That's pretty much the nomenclature of the protein molecules, amino acids attached to each other. Carboxyl end and amino end. So when you attach 2 amino acids together, one end of the amino acids right here, my pinky finger, is an amino end, and the other end right here is a carboxyl end, acid end. That's true for all protein molecules. Yes? If you don't believe me, let's look at them right here. So you have 1 amino acids right here, the R group, central carbon, amino group, an acid end right here attaches to another amino acid right here. So this is called a dipeptide, and the bond between them, this blue line, as you can see, the bond between them is called a peptide bond, which is a covalent bond, not James Bond. It's not James Bond. It's a peptide bond, it's a covalent bond. A peptide bond, it means it's a covalent bond which is attached between 2 amino acids. Okay? Then another one, when you're attaching this dipeptide to another amino acids, the OH of the acid of this one is combined with the hydrogen of the amino group of that amino acids, and they form what? Water. And then the bond between that guy, that carbon, and that nitrogen, the bond between the carbon and nitrogen, between carbon and nitrogen, is called a peptide bond, which is attached right here. It's called a peptide bond. Quickly, refresh your memory. You have a quiz on it today. When you combine this process is called what? When you add this amino acid to that, it's called what? Huh? Dehydration. If I'm wrong, tell me Amir, you're wrong. She's wrong. It's called dehydration. Is that right? Let's go. So at the end of this diagram, at the end you have a carboxyl group, an acid group. You guys see that? This is an acid group. And this is an what? What is this group? Amino group. This is an amino group. This is an acid group. If this was 100 of them, attached amino acids, then what would be on one end? These are all amino acids. Imagine all of the circles is amino acid. One end is what? COOH, acid group, and the other group is what? NH3, an amino group. Are we making sense? All combined, all amino acids. One end of it, amino group, one end of it is carboxyl group. He's calling it a carboxyl. See, and then those terminologies, they call it N-terminus and C-terminus. Be familiar with those when you grow up and go on and move on to other classes. They use those terms. Be familiar with it. Okay, another animation. Let's go over it. Hey.
Most proteins are folded into a complex globular shape. Each protein molecule consists of one or more chains of amino acid monomers. The amino acids are linked by peptide bonds, so a protein polymer is often called a polypeptide. Because they are so complicated, proteins are usually described in terms of 4 levels of structure.
Okay. The primary structure, there are 4 structures. Primary, I should have bolded these. The primary, secondary, tertiary, and quaternary. Okay, a common essay question in the past I like to ask, what are the differences? Can you give me an example? What are the differences between primary, secondary, tertiary? What the question I asked, is it up there? No, 4 levels of protein structures. I should have asked, what are the 4 levels of protein structures? And then during exam, you guys wondering, what does that mean, Amir? Are you talking about 4 different amino acids? No, I'm talking about 4 different levels of protein structures. Okay, the primary one. Structure of protein is a unique sequence of amino acids. This is a primary protein molecule right here. You're looking at it. That's a primary protein molecule. Then, what is a secondary? If I have a piece of paper right here. Imagine I should cut 2 of these together. Imagine, oh, here we go. Same as that one. Imagine each one of this is an amino acid. This is an amino acid, this is amino acid, this is amino acid, amino acid, amino acid, amino acid, amino acid, and they're connected together by peptide bond, right? Just imagine that. This is a primary structure. They're not folded, they're not twisted. This is a primary structure. But when you have it twisted like this, okay, then this is a secondary structure. When you have the protein, the amino acids, secondary structures found in most consist of coils and fold in the polypeptide chain. That would be a secondary structure. Okay. Then a tertiary structure determines by interactions among various sides chains. So you have of the R groups. So this is a secondary structure. And then a tertiary structure would be like this. It folds back and forth and like this. That would be a tertiary structure. And then quaternary structure would be, you have 2 of these right here. This is another chain of amino acids, right? You have 2 of them, and then twisted or fold. And then now these 2 are bonding together, interacting with each other, then that's a quaternary structure. So now you know the definition of a primary, secondary, and I gave you a picture of it, tertiary and a quaternary. Here is a primary structure, amino acids, just back and forth, back and forth. Insulin would be a good example of a primary structure. I didn't write it down here. Maybe I should have wrote it down somewhere in here. Those of you who have your PowerPoints, write it down. Insulin would be an example of a primary protein molecule. Primary. Okay? Then here they are, secondary, as I showed it to you guys. Then secondary, you could have alpha and beta. I need to talk about that in a minute. I will. A secondary structure is alpha helix. This is amino acids. This is alpha helix right here, twisted in that fashion. That's alpha helix. Then secondary structure also can be pleated. Like I fold them back and forth. Pleated. You all know, have you seen those pleated skirts? You know what I'm talking about? Pleated pants, pleated skirts. You all familiar with that term? What it is? If you don't know what that means, right? Here, this is pleated. Right here. You don't know what I'm talking about? That's pleated. So these amino acids can be twisted like this, alpha helix, or they can be pleated like that. And they both are secondary structures. Twisted, alpha helix, or like a snail, if you would, or pleated beta. They call that a beta, pleated sheet. And then what you have, then that actin would be in muscle cells, would be an example of a beta. Okay, actin, it's in your muscle cells, would be an example of beta. Myoglobin, which is in your muscle cells, stores oxygen for you. Myoglobin is a protein molecule, is in muscle cells, and that would be an example of a tertiary structure. It has both the alpha and the pleated sheets, the beta, alpha and beta. And then of course, quaternary structure is hemoglobin, which is two of these together. You remember that two of these together would be an example of hemoglobin. Hemoglobin, of course, have four polypeptide chain attached. Is there any question?
Why did they fold? What makes the chain do all that?
Okay, so you're asking two separate questions. But the first question is, what good question? The second question was a good question. The first one was good too. But what makes them fold like this or twist like this? It's because of the amino acids right here form a hydrogen bond with the amino acids from the other end. And that's why they become twisted. This chain become twisted like this because this amino acid right here is forming a hydrogen bond with that amino acid right here. Okay, do you see that? It's forming a hydrogen bond right here. It's forming a hydrogen bond between that one and that one. These are forming a hydrogen bond between this amino acid right here and that amino acid. That's why you have pleated or fold. Why? The first question, answering your first question, why is it like this? If you want to have an amino acid this long, it takes a lot of space in the cell. The functionality is not going to be functional. Not everything can be straight chain. So in a cell like this, you have thousands and thousands of different protein molecules. They have to be folded, right? Mm-hmm. So you fold a letter and put it in an envelope. You don't put the letter in the envelope and send it. That would be if we could not fold this and mail it. It would take a lot of space. That's the best answer I have for your first question. Why do they fold? Why do they pleat? Because they take space, just like anything else. The second question you ask is a multiple choice question in the exam. How do they do it? Because of the hydrogen bonds between that amino acid and that amino acid. Between this amino acid and that amino acid, they form a hydrogen bond or a disulfide bond. That's another possibility. Sometimes they can form a covalent bond between the amino acids. Here we go.
Each protein has a unique primary structure, a particular number and sequence of amino acids making up the polypeptide chain. 20 different amino acids are used to build proteins. Theoretically, the various amino acids could be linked in almost any sequence, forming an almost infinite variety of different proteins. This illustration shows some of the amino acids making up the primary structure of a protein. The structure of a single generalized amino acid is shown below. The main backbone of every amino acid is the same. This is what forms the backbone of the polypeptide chain. It is the R group, which projects out from the backbone, that makes each of the 20 kinds of amino acids unique. Different amino acids have different properties that affect the folding of a protein. Thus, primary structure ultimately determines the shape of a protein, which determines its function.
Okay. Every protein molecule has a different shape, and of course they have a different function. The coils and folds of secondary structure results form hydrogen bonds between repeating constituent of the polypeptide backbone. Typical secondary structure are a coil called a helix or a fold, or they are pleated called beta and alpha helix. Okay, alpha helix and beta. Secondary structure, again, I'm going over it. Am I going backward? No. Okay, secondary animation. Let's go over what I thought we talked about.
In most proteins, parts of the polypeptide chain are coiled or folded, forming twists and corrugations. This is secondary structure. The turns and folds of secondary structure contribute to the protein's overall shape. One kind of secondary structure is the alpha helix, where the chain.
For example, this is a alpha helix. This is the beta, right? What is in between? That's another alpha helix. What is in between? What do you think that is? What do you think that is? A primary. Yeah, it's just straight chain of amino acids. They're not folded. They're not pleated. It's just straight chain. It's a primary. Oh, hey. Sorry.
In most proteins, parts of the polypeptide chain are coiled or folded, forming twists and corrugations. This is secondary structure. The turns and folds of secondary structure contribute to the protein's overall shape. One kind of secondary structure is the alpha helix, where the chain twists. Another is the pleated sheet, where the chain folds back on itself, or where two regions of the chain lie parallel to one another. Secondary structure results from hydrogen bonding between atoms along the polypeptide backbone. Oxygen and nitrogen atoms along the backbone are highly electronegative, giving them partial negative charges and leaving nearby hydrogen atoms with partial positive charges. These negatively and positively charged atoms are attracted to one another at regular intervals along the chain, causing parts of the protein to twist or fold back upon itself.
Okay, Reno, I answered your question. Let's see what this one is. An idealized alpha helix. If you look at this one again, one more time, the amino acids are sticking out. This is alpha helix. Look at them. Some of the amino acids are sticking out. This is the one that all of the amino acids are inside. Okay, nothing sticking out. So we have different ones, different type of. Again, this is a pleated one, and this is the another pleated one, which is showing with stick model. Tertiary structure. Let's go over tertiary.
Superimposed on primary and secondary structure is tertiary structure, irregular loops and folds that give the protein its overall three-dimensional shape. The irregular folding of tertiary structure results from interactions among the R groups of amino acids. Acidic and basic R groups ionize, and these positively and negatively charged groups may form ionic bonds. Polar forces also contribute to tertiary structure. hydrophilic or polar R groups may hydrogen bond with one another or turn outward and hydrogen bond with the surrounding water. Hydrophobic, nonpolar R groups cluster on the inside of the protein away from water. Tertiary structure may be further stabilized by strong covalent bonds between sulfur atoms in certain R groups.
Okay, tertiary, the overall shape of a polypeptide results from interactions between R groups rather than interactions between backbone constituents. That's another difference of the tertiary structure. Okay, these interactions include hydrogen bonds, ionic bonds, hydrophobic interactions, and van der Waals interactions. I never talked about van der Waals interactions. It's just like it's an attraction between two molecules, which is just like hydrogen bonds. Again, I'm not, do not worry about it. Strong covalent bonds called disulfide bond. Yes, you have to worry about that one. We talked about it. Disulfide bridges may reinforce the protein's structures. Again, as I said, in your hair, if your hair is straight, then you do not have much disulfide bond. If your hair is curly, you have more disulfide bond. And I talked about that. Okay, here they are showing you all of different type of interactions between that amino acid and that amino acids. So that would be hydrophobic interactions, van der Waals interactions. This would be ionic bonding, type of ionic bonding. This is a disulfide bond. This is a cysteine. This is a cysteine. None of them is methionine. Methionine does not form disulfide bond. Okay, methionine is another amino acid that has sulfur. We talked about that. Is that right? Yeah, quaternary structure and the last one.
Some proteins consist of 2 or more polypeptide chains.
Here.
The fourth level of protein structure, quaternary structure, results from the combination of 2 or more polypeptide subunits. Quaternary structure is stabilized by the same sorts of attractions that stabilize tertiary structure.
An example of this is hemoglobin.
Hemoglobin, the red oxygen-carrying protein of blood, is an example of a protein with quaternary structure. It consists of 2 kinds of polypeptide chains. 2 of each, a total of 4 chains, make up each hemoglobin molecule.
Okay? And quaternary structure results when 2 or more polypeptide chains form one macromolecule. Collagen, you have them under your skin, you have them in your joints. It's a very common protein molecule in your body. That's the name of a protein molecule, collagen. The name of the protein molecule on the surface of your skin is called keratin. So protein molecules have a name. Keratin, collagen under your skin, in your joints. Okay, so collagen is a fibrous protein consisting of 3 polypeptides coiled like a rope. And hemoglobin is a globular protein consisting of 4 polypeptides, and 2 alpha and 2 beta, which are different than, this is the structure of a collagen. And of course, this is hemoglobin, which has 4 polypeptide chain, beta, beta, alpha, and alpha. So these are subunits. A beta and alpha subunits is different than beta and alpha structure that you're learning here. Here they are, again, sickle cell and normal red blood cells, normal protein molecule. Another name that you should be familiar with it is called denaturation. What is denaturation? The best example of denaturation I have, if you fried your egg this morning, you took an egg and you fried it, you denature those protein molecules in the egg. Can you go back to the original shape? No. There's no way after you frying it, you can go back. There are 3 or 4 factors that cause denaturation. One of them is heat. Denaturation can be caused by heat. The other one is by strong acid. You expose the protein molecule to a very strong acid, you denature it. And then the third one is strong base. So those are the 3 things can cause denaturation. However, in Mother Nature, denatured proteins in Mother Nature, I didn't say outside of environment, when you heat it or you put a strong acid to it or you put a strong base to it, it can renature. In Mother Nature, protein molecules can be renatured. Okay, that happens inside of the cells all the time. So denaturation and renaturation occurs in cells. Proteins folding in the cell, it is hard to predict a protein structure's form. It's primary. Most proteins probably go through several stages of their way to stable structures. Chaperonian are protein molecules that assist the proper folding of other proteins. Diseases such as Alzheimer, Parkinson, and the mad cow disease are associated with misfolding. So you have all of these proteins inside of the cell, all of the proteins inside of the cell, they have to go to another protein to be folded. Just folding by itself does not happen. Folding by itself does not happen automatically. They go to another protein. This primary amino acids will go to another protein which is called chaperone right here. They go to another protein molecule, and then this protein molecule, chaperone, will fold them. Am I making some sense? So you do need another protein molecule to fold the primary. Look at the polypeptide. This is just polypeptide right here going into unfolded polypeptide into a cylinder form. This is called chaperone right here. And then it has a cap. Goes the cap, and then the cap opens up, the folded protein comes out. I hope I'm making some sense. And all of that happens inside of a cell. We'll talk about that. It happens inside of Golgi apparatus, right here. Golgi apparatus, you will learn that later on. Or endoplasmic reticulum. Rough endoplasmic reticulum, of course. You will see that. Okay, let's go to nucleic acid. All done with proteins. All done with proteins. Nucleic acid should not take long, 10 minutes, 15 minutes. Your textbook talked about more details than what I would like to talk about it. I will talk about it for 10, 15 minutes and then you're done. Okay, nucleic acid store, transmit, and of course help express hereditary information, the information that you get, the DNA that you get from your mom and dad. Of course, half of you is from your mom and half of you is from your dad. So that they transmitted the information, the inheritance information to you guys. And of course, that information from half of you from your mom, half of you from your dad is translated to either you have brown eyes or blue eyes. That's the information. The brown eye, blue eye is a protein molecule. Brown eye, blue eye is protein molecule. But who orders you have brown eye or blue eye? Who orders you have blonde hair or dark hair? The DNA. Okay, the DNA orders that you have brown eye or blue eye. The amino acid sequence of a polypeptide is programmed by a unit of inheritance called a gene. So you do have, I wish we had a model of DNA, but you do have, can I erase this quickly? A gene, we will talk about this for rest of semester, something you need to know for rest of semester. What is a gene? A gene is a sequence of, a gene right here is DNA, that's DNA. Okay, a gene is a sequence of DNA, nucleic acid, that eventually tells the cell what protein molecules to make. That's a gene. Your gene determines how tall you are. Your gene determines how short you are. Okay, so those are all determined by your genes. Genes consist of DNA and nucleic acid. So this from, if I take this off right here, this is two strands. One strand of it is called nucleic acid. This is two of them. Am I making some sense? This is one, this is two. So I just draw the second one for you, if you would. So that is called a nucleic acid, made up of monomers called nucleotides. So this would be a monomer. This would be another monomer. This is another monomer. This is another monomer. Am I making some sense? Together is called a polymer. And then this polymer is called nucleic acid. It's not called protein. This polymer is called nucleic acid. Before you learned it was called glycogen. You remember that glycogen is a polymer? Cellulose is a polymer. Did you study your notes every night from beginning to the end? Are you meditating every day? Don't say, Amir, you never said that stuff to us. I'm telling you how to succeed in life, in this class, in everything you do for the rest of your life. The rest is up to you. Just like my family members, I tell them what to do, they don't do it. All I can do, lower my lower lips. Right? What else can I do? I told you so. You don't do it, it's up to you. It's your life. Okay, so anyhow, there are two types of nucleic acids, DNA and RNA. Nucleic acid, the long chain, there are two types of it, DNA or RNA. RNA could be one strand. This is DNA. DNA is double-stranded. There are other differences. RNA is single-stranded. RNA is single-stranded, DNA is double-stranded. We will go over the details of DNA, RNA, and study that a little bit more in detail, a lot more in detail in exam number 3. And of course, portion of 4, I'm sorry, there is no 5. So, DNA stands for deoxyribose nucleic acid. That's where the term DNA comes from. D-N-A. Do you see that? Deoxy. It means, deoxy means without oxygen. This ribose molecule does not have oxygen. You will see that in future PowerPoints. Ribonucleic acid, RNA, has the ribose, which is a sugar molecule, has oxygen. That's the difference between DNA and RNA. That's another difference, but you will learn that. Here is, of course, inside of the cell, right here, inside of the cell you have DNA, and DNA makes, of course, mRNA, and then we'll talk about that. RNA makes RNA, RNA goes to the cytoplasm, goes through ribosome, ribosome is a protein molecule, and of course makes another protein molecule. We'll talk about this later on. Okay, so you don't have to worry about it too much. The components of nucleic acids. The nucleic acids are polymers called polynucleotides. Polynucleotides, right here, right there. And then each polynucleotide is made up of monomer called nucleotides. So that one monomer is called nucleotide. This is called nucleotide. This is another nucleotide. This is another nucleotide. This is another nucleotide. Am I making some sense a little bit? And then each nucleotide consists of a nitrogenous base, a pentose sugar. Pentose means what? Anybody? From 5, a sugar molecule that has 5 sides. This is a pentagon. It's not a hexagon. Hexagon was what? Something you're going to draw these for me on Wednesday. Bring your pencils and a pencil sharpener and eraser on Wednesday. Of course, you need it for exam, but you're going to draw a lot of these for me on Wednesday and hand it in. Okay, so each polynucleotide is made up of monomers called nucleotide. Each nucleotide consists of a nitrogenous base and a pentose sugar and you will see that, a phosphate group. The portion, is that right, of a nucleotide without the phosphate group is called a nucleoside. If you have, what he's saying, if you have a sugar molecule like this, and the nitrogenous base, which I will draw it for you guys in a minute. If you have this together, this is equal to a nucleoside. Do it make sense? If you have a pentose sugar,
Horrible.
you have a pentose sugar, then you have nitrogenous base. Can I draw it? NB, nitrogenous base, and then you have a phosphate. Then this is called nucleotide. Nucleotide and nucleoside. Side does not have phosphate. Nucleotide has phosphate. That's the difference. Later on, you will see that. So it's you better be familiar with it until we get to it. Nucleoside, nitrogenous base plus sugar. There are two families of nitrogenous base: pyrimidines, which is cytosine, thymine, and uracil, have a single 6-membered ring. You will see a picture of it. I'll talk about it. Purines, adenine, and guanine have a 6-membered ring fused to form a 5-member ring. Easy to memorize this, guys. Easy to memorize this for the rest of your life. That's how you remember it. The way I say pyrimidine is a longer word, right? It has 1 ring. Purine is a shorter word. It has 2 rings. It's the opposite. A normal way of naming it in the past, pyrimidine has a longer word, supposed to have 2 rings, but no, it has 1 ring, and purine, a shorter word, has 2 rings. And now you will see that. In DNA, the sugar is deoxyribose, in RNA is ribose, nucleotide is nucleoside plus phosphate group. We already said that. Here we go. Okay. Here is a pyrimidine. There are 3 pyrimidine. There are 3 of them. And uracil is found in RNA. Uracil is not found in DNA. DNA does not have uracil. RNA has uracil. Cytosine and thymine, they both are single ring. And look at the name. That name is longer, right? That name is longer, so it has 1 ring, 1 ring, 1 ring. And they call them nitrogenous base because look at all of these nitrogens. Look at all of these nitrogens. They call them nitrogenous base. Of course, they are base. The pH level of it is a base. It's not an acid. So they call them nitrogenous base. And then purines. Look at purines. The name is shorter than that name. They have 2 rings. 1 ring and another ring. 1 ring, another ring. And then there are only 2 of them. Adenine and guanine. Adenine and guanine. Sugar molecule, here is a deoxyribose. Look at that sugar number 2. Carbon number 2, I'm sorry. Carbon number 2, the H does not have oxygen, but look at the number 2, the carbon number 2, it has oxygen. It has oxygen. So this is ribose, 5-sided sugar molecule. I did draw that for you. 5-sided sugar molecule. Right here, carbon number 2 does not have oxygen. So this is found in DNA, and this is found in RNA. The way they are arranged together, both DNA and RNA, you have a nitrogenous base, right? One of these 2 guys, nitrogenous base attached to a sugar molecule is attached to a phosphate. Nitrogenous base, which is a pyrimidine, attached to a sugar molecule, attached to a phosphate. Again, same thing, they're being repeated and repeated and repeated and repeated.